For her postdoctoral work, Carsen moved to Janelia Research Campus to work with Marius Pakitsuru and Carl Svoboda. Did I get that wrong? I think I did.
It was pretty close. It was good.
Okay. Sorry. Last year, she started her own lab at Janelia as a group leader. In addition to answering important, interesting scientific questions, some of which she'll tell us about today, Carsen has also made great contributions to neuroscience by developing widely used analysis software, such as Sweet2P and CellPose, that many of us use, and also making her data sets widely available. Her lab focuses on complex, high-dimensional cortical computations using recordings of tens of thousands of simultaneously recorded neurons. And today she'll tell us about some of that work in her talk titled High Precision Coding in Visual Cortex. So briefly, just a few procedural things to mention. This meeting is being recorded and streamed on YouTube. If there are questions, please post them in the Q&A box by clicking Ask a Question at the bottom of your screen. If you would like to ask your question in person, just write the words on screen after your question, and audience members can vote for each other's questions throughout the talk. So without further ado, I'll thank Carsen again and let her take it away.
Thanks so much. Thank you so much for inviting me to give a talk here. It's quite an honor. And thanks everyone for coming, particularly the Americans, given the week that we've had. And I guess I just want to say, as a white American and to all the white Americans here, I hope this kind of reinvigorates our efforts for diversity, equity, inclusion as we work towards those things, and as we've been working towards them this year. So I'm going to give a talk today about high precision coding in visual cortex. And for those of you who aren't familiar with the kinds of recordings that we do large scale recordings in mice. So we take mice and we look at their brain. So this is the mouse brain, and we make these small windows into their skull. So these small few millimeter windows, and we're able to record neurons through these windows using a technique called calcium imaging. So we put a microscope and we're able to record these neurons, which are flashing whenever they fire. So when calcium comes into the cell, these neurons are flashing, and that's how we record their firing. And so rather than just recording that small field of view, we end up zooming out and also recording many planes simultaneously, and that allows us to record many thousands of neurons at a time. And here's an example of what one of those recordings looks like. So each of these squares here is a different plane of this recording, and in the upper left is the image that's going to be shown to the screen on a screen in front of the mouse. And at the bottom, this is the mouse that's head fixed to the microscope and free to run on this ball below the mouse. And so when the images come in, you can see visual cortex lighting up. It responds to images. And then there's also activity in the absence of images. You can see these neurons continuing to fire, fewer neurons, but there's still activity. And so we take these large-scale recordings with many gigabytes of data, and we extract neurons from these recordings in an automated way using a pipeline we've developed called Sweet2P. And each of these little circles here is one of these neurons. And then we take the sum of the pixels in these circles across time, and that gives us these traces here. And then what I'm going to be doing in this talk is we basically bin each of these traces at each stimulus onset and take a few of these bins for each of these stimuli and work with this matrix that's neurons by stimuli. And so my talk is kind of motivated by this idea that often people don't think of rodents as visual animals, but they do use their vision to explore the world. And here's an example of a video of a rodent using vision. This particular rodent is a squirrel, which we don't specifically study, that picks up a GoPro and runs up this tree. You can see it jumps to other branches there. And so at every position here in this tree, it needs to look around and see where these branches are, what angle they're at. And if it miscalculates these angles that it just sees for an instant, then it will fall to the ground and not necessarily survive. So it's important to use to have accurate vision in order to explore the world, particularly in the case of a squirrel. So the question we wanted to ask was, how much information do rodents have in visual cortex, and how can they use this information to guide their behaviors? And the fundamental limiting kind of factor in visual cortex is the fact that the responses of these neurons are noisy. So we want to be able to see the world in high fidelity, but if you look at a single neuron and record from a single neuron in visual cortex, the responses on a single trial are noisy. So we have these images we show to the mice on a screen in front of the mouse, and we show many different orientations. Each of these dots is a single response of this neuron to these different orientations. And we can fit a tuning curve here, but what you can see is there's wide variability around this tuning curve. So these angles here around 330 degrees, you can see there's many different responses that this neuron gives, even though it's a very similar stimulus. And so this is a neuron that has relatively low SNR signal. That's the signal to noise ratio. But we do see neurons in our population which have really good SNR. But this is, for instance, this is the best neuron out of 20,000. So most neurons look like this noisy neuron. And so if I'm a squirrel navigating the world and I need to use this neuron to pay attention to where I'm going, I might have problems. And so this is the distribution of SNR across the population, and on average, most of these neurons look like this neuron here. But as a squirrel or a mouse, I don't only have one neuron, I have many neurons. So ideally, I can average the responses across many neurons in order to get an accurate representation of the external world on a single trial level. And so what we first proposed was that we thought, well, maybe most of the noise is independent single neuron noise that won't affect the coding of the angle. And if that were the case, then we could just average these different neurons. So these are examples of responses of different neurons. We have this true angle here in brown. And if we average these neurons' responses, we get the correct response to this bar here, basically this branch. And so if the noise is independent across neurons, or in another case, if we have correlated noise that's not in the stimulus space, then we're able to average over these neurons and not have impaired coding of this branch that we want to jump to, for instance. And so I'm going to go through this, that we found in our recordings that there is correlated noise, and this is something that people have seen in the past. But we found that this correlated noise that's related to the behaviors that the mouse performs, like running, whisking, and sniffing, they drive correlated activity, but it's orthogonal to the stimulus-driven activity and therefore does not impair coding of stimuli. And we also found that there was 1D gain modulation of neurons by arousal, and this also did not impair coding. And so I'm going to go through this idea that there is correlated noise that's related to behaviors, but I'm going to show you how it's orthogonal to stimulus-driven activity and therefore does not impair the coding of the population to these different stimuli. And so what we found was that these spontaneous behaviors do drive activity. So what do I mean by that? So the mouse is free to run, whisk, sniff while it's on the ball. And we see neural activity related to this running, whisking, and sniffing. So in the upper left corner, this is a video of the mouse's face, and we've decomposed the mouse's face into its principal components, which are shown here. And what I hope will be evident in the video is that you'll see activity of these neurons on the right simultaneously with this. And particularly when the mouse starts running here, you'll see this increase in activity. So there's activity across visual cortex that's correlated to the behaviors that the mouse is doing, not just stimuli coming in. So this is, for instance, in the absence of stimuli. And what we found in this paper, I'm obviously going through this in a very abbreviated way, is that we were able to take neural activity and predict it from these behaviors that the mouse is performing. So this is a raster of neurons by time. And this is, again, the same raster of neurons by time where I've replaced every single row here with the prediction from the behaviors that the mouse is performing at every single time point. And what's hopefully evident, so we want these to be as similar as possible, and what's hopefully evident is that there's large groups of neurons across the brain that are able to be predicted by kind of the behaviors of the mouse that have this correlated activity. And so this is in the absence of stimuli. There's these neurons that are driven by running, whisking, sniffing, grooming, these other things. And this is correlated across neurons. So this is correlated noise. So the question is this going to impair the coding of these visual stimuli that the mouse is also seeing? And so what we found was that this is not, in fact, a problem. So I'm going to go through this idea of the orthogonality of the representation of these spontaneous behaviors versus these visual stimulus representations with kind of a toy example. So we have visual stimuli. We have the same visual stimulus, for instance, let's say we show it twice, the same oriented grading. And I'm going to take a single neuron here that's correlated to running. And so when the mouse starts running, this neuron's activity goes up. And then when the stimulus comes in, this neuron's also driven by the stimulus, its activity goes up. And then the mouse is not running anymore, and this neuron still responds to the stimulus though. So if I had a single neuron in this population and I was trying to figure out what stimulus was being shown, I'm going to have a hard time because this neuron is also modulated by the running of the mouse. So on a trial-by-trial basis, there's this noise that we've been talking about. But in the brain, we're recording, there's many neurons, and I can, for instance, take a neuron. There are neurons in the population that are anti-correlated to running. So here's an example of a neuron that's anti-correlated with running. Its activity goes down, and then there's still this input there. It's activated by the stimulus, and then it's activated by the stimulus again. And so we have, as long as we have neurons in the population that are driven by the same stimulus that are correlated and anti-correlated with these behaviors, we can average across these neurons and get the correct response to the stimulus, or a consistent response across trials. And so this is the average neural activity across these two neurons. And because there's a neuron that's correlated and anti-correlated, we get a consistent response from trial to trial in this direction. And so we can generalize this then. We can compute the stimulus vector across all the neurons in the population. So we take the average responses of neurons to this single given stimulus. So neuron 1 and 2, they have on average relatively high responses to the stimulus. So we'll give them a 6 and a 5 in this vector. And then neuron 10,000, we're going to give maybe a size of 3. And what we're going to do is we're going to project this vector across all of the time points in our recording and see how this direction in space varies across time. Is it going to be influenced by whether or not the mouse is behaving or doing other things? And so we're going to take a recording where we have periods where there's no stimuli. This is just the mouse is able to run and whisk and do whatever it wants. And then when there are visual stimuli, and again, the mouse is free to whisk and run and do whatever it wants. And we're going to see how big is the magnitude of this vector, how big is the activity in this direction in space when there's no visual stimuli versus when there are visual stimuli. And so if the neurons are correlated and anti-correlated to behaviors, then what we expect when there's no visual stimuli is that it's going to be flat here. And that's indeed what we saw, was that there's neurons that are correlated and anti-correlated to running, correlated and anti-correlated to whisking, and many other behaviors in this single stimulus vector. And so then when the stimulus does come in, though, this vector is activated by these different visual stimuli that are on the screen, and most activated by its direction that it's most driven by. And we can do this for other visual stimuli, for other different oriented gradings that we showed, and see that in all cases, you see this completely flat curve here. Well, there's no visual stimuli on the screen. So this is, again, this average, it's all of these, this direction in space averages out to 0 activity. And then there's activity in the stimulus space. And then if we look at the, so for instance, if we take the principal components of the activity in the absence of visual stimuli, so these are kind of these behaviorally driven components, we see that they continue, they're sort of ongoing, whether or not there's visual stimuli on the screen, they just continue encoding the behaviors that the mouse is performing. So I'm kind of, let me actually go back. Let me kind of, so what we think is that there's this, what we showed in our previous paper and what we think is largely true in at least in passively viewing mice, is that the stimulus-driven space is orthogonal to the behaviorally driven space. So the noise that we see that is correlated across neurons that's related to behavior does not impair the coding of visual stimuli. So if a squirrel or a mouse is running or not running, they should be able to still perceive, kind of in theory, the same stimulus, because there's neurons that are correlated and anti-correlated to running in this stimulus vector space. So does anyone have any questions about kind of this idea of stimulus and behavior orthogonality, or about how behaviors drive visual activity and anything like that before I go on? You're welcome to ask questions in the chat too. That's totally fine. I can see the chat.
Can I ask a quick question? So my impression was that there were a number of studies that showed that running on that when mice run, there's an overall enhancement in the visual responsiveness of neurons. Is that at odds with this idea at all?
That's a great question. No, it's not. It's actually it's so in the case we think of there being an additive subspace and there also being a multiplicative subspace. So in addition to this additive subspace, there's also this gain modulation on single trials that's actually a different direction that is largely driving more of the neurons in the population, so that they are, when the mouse is running, these the responses of these neurons is on average higher. So let me explain that a little better. Sorry. So there's these gain, each neuron, you can think of, there's this gain modulation that's kind of this, that's correlated with running. And most neurons have a positive weight onto this gain modulation. On average, more neurons have a positive weight onto this gain modulation. And so, so on average, the population activity will go up when the mouse is running and visual stimuli relative to not running. But that's specifically a multiplicative modulation. So that doesn't change, that doesn't have, that direction in space isn't being activated by these behaviors. So when the mouse is doing these behaviors, that's not actually in the stimulus space. That makes sense. That's adding a separate stimulus. That's adding a separate, that's in a different neural vector, where there's neurons that are positively and negatively correlated to running in that vector. Those are different neurons to some extent than neurons that are correlated in the gain modulation. So there could be neurons that are suppressed by running in this case, but might have a positive gain modulation term. That can happen.
Thank you. I think that also addresses Jason's question in the chat, which is, does it matter if the interactions are additive versus multiplicative?
Right. Yeah. So what we found, it does matter, but it depends on in what context. And so the additive effects, because they're orthogonal, we can ignore them. And then multiplicative effects could potentially, what we found was that they're largely one-dimensional. We weren't able to fit a higher dimensional gain coefficient, so it's largely one-dimensional. And that could, in theory, impair coding if it was like a really large effect. So if you had really large gain modulation and neurons were basically like close to zero firing when there's no gain, and going up to and pretty much off when when the gain was off, then you would see an impairment of coding, if that makes sense. So like if neurons had really low activity and then the gain modulation was largely what the large percentage of the stimulus response, then we'd have a problem. But that's not what we see. We don't see that neurons are just like completely off if there's no running, if that makes sense. Mm-hmm. And so in a supplementary figure in the bioRxiv paper on high precision coding, we have a supplementary figure where we try to decode including a gain modulation term, and we don't see significant improvement in the decoder, suggesting that it's not a big influence in terms of decoding, that the neurons are still firing even if the mouse is running or not running, if that makes sense.
So there's one more question in the Q&A, and then maybe we can kind of let you proceed. So Jag Conwell is asking, isn't negative correlation also contributing to encoding activity in response to the stimulus? So why does the vector show a flat line?
Yeah, so there are negatively correlated and positively correlated. So there are neurons that increase their activity and decrease their activity with running and with whisking and these other things, that they might have some firing rate, and then that firing rate's suppressed when the mouse is running, for instance. I don't know if that answers the question. I don't think I can see it, but. There's another question from Arrow. It seems surprising that the relevant or relevant signals can be pulled out or canceled just using linear operations. and the question of, do you think these aspects of information are encoded linearly? Yeah, that would be the suggestion, that largely it's kind of subsets of neurons that are just driven by behavior and maybe don't necessarily have strong recurrent connections that are creating these nonlinear effects. But we don't really know. Our best fit for a behavior to visual cortical activity model is linear. So that does suggest that it's that's currently the best model, that this recurrent activity might not be so large in that subspace, but we don't really know. But if everything is linear, it's not so surprising that these things are orthogonal, because you're in a really high dimensional space that's 10,000 dimensional. So the probability that two random vectors are not orthogonal is actually kind of low in that sense. Was there any DC removal? Yeah, we always subtract off the mean activity. Yes. Sorry. So no, I think all the questions are really good. So keep asking questions. Yeah. So what I hope kind of came across in all of that was this idea that us and others for a long time have seen noise when visual stimuli are shown to mice or other animals. But this noise isn't necessarily in the stimulus space. It could be correlated across neurons, but it could be related to the behaviors that the mouse is performing, for instance. And that behaviorally driven activity is orthogonal to the stimulus space. So we don't need to worry about it when we're building a decoder or in terms of in limiting information. And so the next question we asked was, is there other noise that might limit information? Is there noise that's actually in the stimulus space, unlike this behaviorally driven activity? So our first hypothesis is that no, it's just all independent noise. And so what we did was we built an independent decoder to see if an independent decoder can perfectly decode these angles that we showed to the mice, these different oriented gratings that we showed to the mice. So we built an independent decoder where every neuron, we fit a tuning curve to each neuron. So here's 3 example neurons. And so you can see these tuning curves fit. And then on a single trial, we look at the response of that neuron on a given trial, and we compute the log likelihood of each angle with respect to that response. And so what you could see, for instance, for this neuron has a high, the tuning curve is high around this angle, and the response of this neuron on this given trial is high. So the log likelihood predicts that the angle is around the peak of the tuning curve. And then, for instance, for this neuron, you can see this in general, there's not a strong, the SNR is low, but in the case of of this response here, it's below the tuning curve. So it suggests actually, it thinks that it might be this angle, but it has a relatively low probability overall because the SNR of this neuron is low. And so it's kind of mostly a flat curve. And then for this neuron, we have, for instance, it has a high response on this trial, and that's also where the tuning curve is high. So we're going to predict that it's likely this angle, but it could also be this angle here. If you see, this is a doubly peaked tuning curve, so it has 2 peaks in its log likelihood. And this is an independent decoder, so we compute each of these log likelihoods from each neuron independently and sum them to get this single average log likelihood over the whole population, and take the peak of this average log likelihood curve to get the decoded angle or predicted angle. And if we do this, we've shown many different stimuli. This is the true angle of all those stimuli, and this is the decoded angle. We do a relatively good job of predicting them. They're largely on the unity line, which is where perfect prediction is. And we look at a histogram of those errors. The errors are around 2.3 degrees, which is not too bad. And on average across recordings, it's around 2.3 degrees as well. And so the next question we wanted to know was, is this decoding error correlated across the population? So we showed that that we can decode to some extent with an independent decoder, but we wanted to know if there are correlations across neurons that are contributing to this error. So what we did was we split the population into 2 halves, and we looked at what the decoder said for each of those 2 halves. And if we look at, for instance, the log likelihood of population 1 versus population 2, we plot them on top of each other, we get a curve like this. And if we zoom into this curve, you can see that these curves are both centered to the left of, I'm sorry, I should have said this, the true angle is this dotted line here. And so you can see that they're both centered to the left here. So there's a decoding error in each of these cases that's around -3 or 4 degrees. And so these are two separate populations of neurons. But despite that, they're randomly chosen. They have correlated errors. And so what this suggests is that there's correlations in the stimulus space. And so I should show the full plot first. So we have population 1 versus population 2, and each of these dots is a single stimulus presentation. And you can see that it's correlated across all these different presentations. And what this suggests is that the neurons have correlations that are in the stimulus space. And so there are some correlations in the stimulus space. So it's not just an independent decoder that's going to be able to resolve perfectly this angle. So I'm going to put an X through this. It's not just independent single neuron noise. There is noise that's correlated in the stimulus space. And in most cases, though, this noise in the stimulus space is fine. So if, for instance, only a subset of neurons are correlated to each other, we can basically, if we know that they're always correlated to each other trial to trial, then we can basically correct for them by saying, okay, we always know these neurons are correlated to each other, and if we take that into account, we'll get the correct angle. And this is kind of like a whitening operation. This is what standard linear decoders do. They're able to obtain the correct angle if only a part of the population is correlated. And so basically the squirrel would be able to jump fine to that branch if we only have subpopulations that are correlated. But there's a possibility that there are information limiting correlations in visual cortex, and this is something that was proposed by this paper in 2014 by Moreno-Bote, that instead of just a subpopulation of neurons being kind of correlated and wrong together, the whole population is wrong together and predicts the wrong angle. And so in this case, there's no way to recover the true angle because all the neurons are saying basically are coding for the wrong angle on a given trial. So we can't recover the information. And so that's why these are called information limiting correlations. And so we wanted to distinguish between these two possibilities, that there are just correlations in the subspace we can correct, in the stimulus space that we can correct for, or are there these information limiting correlations, and ultimately are rodents limited in the amount of information they get from the visual world?
So Carsen, you have a question from Ken Harris in the Q&A. And I think it refers to the previous slide. Apologies. And so the question is, interesting that the wiggles of the curves in the top plot are so similar. Do you have an interpretation of that? It's easy to go back.
Oh, across the whole curve? I guess he means this curve. Yeah, I guess I guess it suggests that that's kind of the way that the noise was on that given trial, that the neurons that were correlated to each other on that given trial had specific responses in that way. There are biases in the population in terms of there are more neurons that will code for horizontal or vertical bars, depending on if it's drifting or static grading. So that will contribute to some of this as well. But I'm not totally sure what it is exactly on this. Yeah, it shows how the noise must have a lot of structure across the population. Yeah, so there is some structure in the noise. Yes, that's true. And that is, we do exploit the fact that there is structure in order to decode better, basically. If you know the structure of the noise, you can cancel it out. Yeah, okay. So then we wanted to distinguish between these two hypotheses. Kenneth is still asking questions. Do the wiggles look different on different trials? Probably yes, but I'd have to look at that. There are several complex tuning curves too across. There are neurons with like more complicated tuning curves. So yeah, I'm not sure. Okay. So how could this information limiting noise arise? So I'm distinguishing between these two hypotheses, and one of them might sound like a little silly to you if you haven't heard it before, that the fact that this idea that the whole population is changing its response on every given trial. But there are some hypotheses for how that noise might arise. So for instance, if there's noise in the retina or noise before visual cortex, like if the thalamus or retina are noisy, then this information will be lost before it even gets to visual cortex. So all the neurons will be wrong in the same way if there's noise there. There's another hypothesis which is somewhat common in the field, is that the neural space is in an attractor state, so that any kind of noise in the neural space moves you along this attractor. So like noise that's outside of an attractor just pushes you back to an attractor. So, and then you just get pushed along this attractor, where the maybe if this attractor is in angle space, then you change the angle that you are encoding, basically, and your noise will never be orthogonal to the stimulus space then. And so yeah, so that's another that's a dynamical system where you would have information limiting correlations as well. All right, so let's see if there are information limiting correlations in visual cortex. So we built a linear decoder to get rid of these correlations in the stimulus space and see if we can indeed get rid of them. And so we took these neurons and we built a linear regression model that we regularized by using these things we called superneurons, where we have these superneurons which have these perfect tuning curves at several angles along 360 degrees. And then whichever superneuron responds highest, that's the angle that is the angle that's predicted by the decoder. And so, oh, sorry. And so if we look at the true angle versus the decoded angle for this linear decoder, we see again that the points are close to the unity line for these many, these 1,000 or so test trials. And if we look at the histogram of errors, we can see that now it's closer to 1 degree error. So rather than 2.3 for the independent decoder. So we're doing over twice better with this linear decoder. So this again reiterates this point that there are correlations in the stimulus space, but we can take them into account we can take into account a large fraction of them using a linear decoder. Now the question becomes, okay, what if we increase the number of neurons and the number of trials? Can we do even better than this? Can we do better than 1 degree decoding error? Maybe what's the kind of limiting factor in terms of decoding these stimuli? And so we looked at the error as a function of the number of neurons and the number of trials. So we can take this decoding error and take subsets of neurons from our whole population and just decode with those subsets and see how that decays as a function of the number of neurons. And similarly, we can see how the decoding error decays as a function of the number of trials. And both of them do decay with the number of neurons and the number of trials. And we can fit a curve to this decaying function. And what we found was that this asymptote of this curve is around 0.7 for neurons. And then for trials, it was around 0.38. So that suggests that if you add more trials, you're more likely to decrease your decoding error even more, suggesting that if you only have a few trials, you're more likely to be overfitting to those trials. It actually might help to have more trials. And that's actually something we can control. We can actually increase the number of trials. We can't increase the number of neurons we record. So that's what we ended up doing. We wanted to see if we increase the number of trials, can we decrease our error even further below 1 degree? And so the way we increase the number of trials is we change our task from this decoding from 0 to 360 to a discrimination task where we're only going to show angles between 43 and 47 degrees and say, is the angle greater than 45 degrees or not? So we have trials that are at 43 degrees, for instance, 46 degrees, and the decoder has to tell if it's greater than or less than 45 degrees. And I'll let you look at these two angles to kind of see how potentially difficult this task is as a human. And indeed, if you are a human doing this task, if you're trained to do it, then you can get a discrimination threshold around 1 degree, which means that 75% of the time, if the angle is 46 degrees, you can tell that it's greater than 45 degrees. But you do have to be trained to be able to do that well. So let's see, how well do the mouse neurons do on this task? And so we build a decoder from the mouse neurons in visual cortex, and we look at the and we look at this curve of performance, what we see is the discrimination threshold is around 0.34 degrees. So 75% of the time, this decoder from the mouse neurons can distinguish 45.3 degrees from 45 degrees, which is a very small difference in angle. And as we look at the asymptotics, it suggests that this alpha, our asymptote here, our y-intercept is 0.1. So it suggests that this could go down even further to 0.1 degrees. And so we're very close to 0 at this point, in a sense, which suggests that we did find that there were correlations in the stimulus space, but we were able to get rid of them. And we don't think there were any information limiting correlations. We're at such a small discrimination threshold that we think that we've basically that we don't need to worry about these information limiting correlations if they do exist. That we're at a very high fidelity coding of these orientations with mouse visual cortex. And what we found was that, or what others have found, is that the mouse performance is much worse than the neural performance. So we found this discrimination threshold of around 0.34 degrees, whereas if you ask a mouse to do this task, their discrimination threshold is around 29 degrees, around 10 to 30 degrees, depending on what paper you kind of look at. And so this is much, much worse than the mouse's discrimination threshold. And so the next thing we did was kind of look into why that might be the case. Does anyone have any questions before I continue? All right. How are you cross-validating? Yeah, we split our trials into train and test trials, and all of these performances I'm showing here are just on the test trials. So we do about a 75% to 25% split. Okay, and in this case as well, it's also a linear decoder. All right, so why is this behavioral performance worse than neural performance? And so first I'll say this again, the neurons have a high fidelity representation of the world, but the mouse might not be doing as well. And yeah, the 29 degrees threshold is on similar lab tasks where the mouse has to discriminate whether a stimulus is greater than or less than a reference angle, for instance. Which is similar in this case. In our case, we have a reference angle, and we see if we can decode whether an angle is above or below a reference angle. All right, so one hypothesis would be that this information is locked away in V1 and it's not leaving V1, so that's why the mice can't do so well on these orientation discrimination tasks. Any idea of a threshold in the wild? All right, yeah, let's get to that later, but that's a good question. All right, so is the information locked away in V1? We looked at if higher order areas have information about the stimulus. And indeed, we found that the discrimination threshold was around 0.37 degrees, similar to primary visual cortex. So this information about orientation at is represented in a highly precise way, also in higher visual areas. Maybe we didn't use the right stimulus. That's another hypothesis. So we showed these stimuli. I'm showing this again because I haven't shown this plot before. Before I was showing linear decoding from all 360 degree angles. But we can also represent this as a discrimination task as well, where we choose a given angle and we look at the discrimination performance. So this is again for the original data I showed from 0 to 360. So that's around a 1 degree discrimination threshold. It's not as good as when we had only 4 degrees, just FYI. But we use these angles from 0 to 360. We showed many different stimuli with the from 0 to 360 degrees. So we showed shorter stimuli. So these stimuli were shown for 750 milliseconds. These were only shown for 100 milliseconds. We got a relatively similar discrimination threshold. slightly worse, but still not on the range of the behavioral performance discrimination threshold. We showed stimuli with random phases. We showed smaller stimuli. We showed complex stimuli like this Minnie Mouse. We showed drifting gratings. We showed drifting gratings with contrast, and we showed drifting gratings drifting gratings with low contrast and drifting gratings with low contrast and with noise. And in each of these cases, the discrimination threshold was around 1 to 2 degrees. And this was surprising, particularly this one. If you watch this stimulus, it's really hard to tell what the angle was. So it was pretty impressive to me that that this information was in visual cortex. There's a question here that I do actually get this question a lot. So is there a spatial representation of orientation given the large population of neurons that are being used for encoding orientation? The answer is we haven't looked specifically in our data, but we largely don't see it at a population level that there's this kind of structure of orientation across visual cortex, or at least it's not as pronounced as in the case of monkey or ferret or that sort of thing. So that is a case where, so in the monkey brain, you have these orientation columns where the orientations are nicely kind of aligned for you across the brain. But that's not the case in the mouse brain. You kind of have the salt and pepper orientation map. And so that might make it harder for the mouse to be able to do the discrimination in a sense than a monkey or a ferret or that sort of thing, or a human. But yeah, but regardless, there's still this very precise coding of orientation, even though there isn't this orientation map. How big are the receptive fields relative to the stimulus stripes? So the receptive fields are closer to the size of this local stimulus. That's about how big they are. So each receptive field will likely be over 2 stripes in a sense, if that makes sense. With the spatial frequency that we showed. All right, so the next question we asked was, all right, so we don't think that changing the stimulus, that we didn't show the right stimulus, that our stimuli were necessarily different from stimuli that experimentalists had used in other papers, that's the difference. We thought, all right, well, then maybe the difference is the behavioral effects. Maybe the state of the mouse is different when the mouse is doing a task versus not doing a task, or running versus not running. So we could look at our data and see if the mouse is running versus not running. What is the discrimination threshold, and does it change? And what we found was the discrimination threshold was slightly lower when the mouse was running, but not substantially. So this is still, and again, this is all passive. So the next question we wanted to ask was, what happens if you're actually in a decision-making task? Are there relationships between behavior and neural variability? And that's what I'm going to go through next. So let me kind of reiterate that. So we've been showing that there's very low neural variability, but in these tasks, people see that the mouse isn't performing so well. So there's a question of if these neurons correlated responses or their noise, if that's in some way influencing the behavior that the mouse is doing. If downstream there's some way that these neurons are influencing the choice of the mouse. And so the next thing we looked at was, is there choice information in primary visual areas? Is this neural variability correlated to choice? So that's what we looked at next. And to do that, we studied Nick Steinmetz's data sets from his paper, his nice paper from 2019. He uses the IBL task, which some of you might be familiar with, which is this contrast comparison task, where there's stimuli that are on the left and the right screen that the mouse has to look at, and the mouse has to choose which stimulus has higher contrast, and then turn the wheel to that direction in order to report the choice of right or left. And so the mouse will, in this case, contrast is greater on the right, so the mouse needs to report a right choice. And so Nick did recordings from around 30,000 neurons when the mice were doing this task. And if you look at the behavior of these so we wanted to know, is this task information limited first? That was the first question we asked, because we do see that in this task is slightly different than an orientation discrimination task. We don't have data for that, but we wanted to know if we can still decode these stimuli in a high fidelity way from this type of task. And so we do know that the behavior in a sense is limited. So if the mouse is asked to do this task and and there's a contrast on the right, for instance, so these are the different contrasts. These are contrast left and contrast right. So if the contrast is 1 on the right and it's 0 on the left, the mouse should always say right. That means that there's a full contrast there, and the mouse is very good at it. Its error is around 2%. So that's this low error here. And then if the contrast is high on the left versus on the right, so up here, you can see the performance is very good. The mouse has an error rate around 3%. And then for these intermediate contrasts, that's where the mouse has slightly more trouble. So if the contrast is 0.5 on the right versus 0.25 on the left, the mouse has an error around on 23% of the trials. So we wanted to know, first, we just wanted to know, is this behavioral error related to the fact that maybe these stimuli are not encoded in a high fidelity way in visual cortex? So we took our recordings. We have these mesoscopic recordings we can do. So we showed images on the left screen and on the center screen, and we looked at if we could decode from responses on the left or the right or on the center screen, and see if we can do this task like the mice were with just from the neurons. And indeed, I don't think anyone would be too surprised by this. We get a neural error rate close to 0. So there's a high fidelity representation of these stimuli in visual cortex. But the mouse is still making these errors. So we want to know if any kind of variability in visual cortex is leading to these errors that the mouse is making. And so to look at this, we're going to take the correct trials that the mouse does, and we're going to build a decoder to decode contrast left versus contrast right on the correct trials. And then we're going to take a subset of these correct trials. We're going to take those out as test trials and match them to the incorrect trials, and use those as our test to see whether or not there were choice, if activity was, if the direction of the decoder is correlated with the choice of the animal versus the direction, or versus the stimulus. And so in this task, we have left versus right. And on correct trials, if the stimulus is higher on the right, then the mouse goes right. So on correct trials, the choice is always correlated to the stimulus. But then on incorrect trials, that's where we have this mismatch. That's where the stimulus is on the right, but then the choice is the left. So we want to see if there's activity that's correlated in visual cortex with this choice information rather than this visual information. So what we did was we took these test trials, these incorrect and correct match trials, and looked at the decoder prediction from these incorrect and correct match trials, and saw if they changed depending on whether it was a right correct choice or a right incorrect choice. So if we build the decoder based on the stimulus period, what we see is that the prediction is very similar for a right correct choice and a right incorrect choice, which suggests that there's not choice information there, that it's largely stimulus information. And then if we train a decoder on the period before movement and even after movement, we can see that consistently the decoder trained on these correct trials is decoding the stimulus and not the choice. So right correct and right incorrect are the same. They're both encoding this stimulus, and then left correct and left incorrect are both encoding the left stimulus, and you can see this here. But then if we look in an area like midbrain, we see something different. So we train a decoder on the first, on the time from this first block when the stimulus is presented. We see that the right correct choice and right incorrect choice have somewhat similar decoded predictions here. But then if we look at, for instance, if we train a decoder on the time from movement, we can see this switch of the decoder prediction. So the decoder is now predicting that the right correct choice and the left incorrect choice are the same. So it's predicting the left incorrect choice is when the mouse moves to the right, even though the stimulus is on the left. And so it's basically encoding the choice of the animal rather than the stimulus. And so this is kind of confusing. Let's kind of summarize this a little more cleanly. So we take the correlation of the stimulus here versus the correlation of the choice of this decoder in each of these different bins. And so in this first bin, what you can see is that largely the decoder prediction is correlated with the choice, that right, correct, and right, incorrect. As long as the stimulus is on the right part of the screen, we can decode this right prediction here. And across all brain areas, we see kind of that they're largely correlated with the stimulus. And then as we move to later bins in the trials, we start to see that these things start to move, that the stimulus correlation starts to decrease a bit, and there's more choice information in each of these areas. They're more correlated with the choice this decoder predicts. And then if we look in the movement aligned case, we see that lots of these brain areas, particularly there's the midbrain there, and other non-visual cortices are more correlated to the choice than to the stimulus. And so what this suggests is that the visual cortex throughout the trials is largely representing, it's largely correlated with the stimuli and not with the choice that the mouse is making. There is some choice-related activity. You can see this correlation here is small, but it is positive. But for the most part, it doesn't seem like neural variability in primary visual areas are correlated to the choice that the mouse is making in this given task. And so we kind of made a schematic of this that basically that there's areas that are largely sensory driven. These are that are correlated with the stimulus rather than the choice. And lots of these areas start out here at the beginning of the trial. They're also representing the sensory information that they got likely from visual cortex, superior colliculus, these other areas. And then they move, their activity moves into being correlated with the choice as the mouse goes through the trial. Their activity is more correlated with what would be motor feedback or motor signals that signal the choice that the mouse is making. And I'm going to skip this part, but this is just suggesting that the way that you learn, whether trial to trial or other strategies, maybe is the difference between performance or not performance, or between the mouse's performance and the neural performance. But yeah, the punchline there is that it might be okay that a mouse can't perfectly do an orientation discrimination task or perfectly do the IBL task, because maybe that's not what a mouse is doing in the real world. Maybe they're exposed to all these different stimuli, and they just need to associate with them once or twice. They don't need this fine discrimination that they're going to do many thousands of times.
Carsen, really quickly,
yeah.
You have a question in the Q&A from Tony Mopshan, which is, does the choice correlated activity look like feedback or attention in primary visual cortex?
Yeah, that's a good question. The feedback is somewhat tuned to the direction, so it's not just arousal, but a large fraction of it is arousal based too. Yeah, in the later it's in these bins here, but this isn't the best analysis to show this. I don't actually have that figure here, but yeah, I get what you're asking. There's a motor feedback, and there is also a modulation by arousal too. So there's another question. Your previous work showed that there's a lot of non-sensory information in V1. Given those findings, isn't it surprising that there is not much choice-related information there? There is choice-related information there. There is a small amount, but it's more, and that, I'm sorry, this figure doesn't really go into that, but there's much more stimulus-driven information there is kind of the point. And the choice-related information that is there is after movement onset, largely. It's when the mouse starts moving, if that makes sense. So that is consistent with our previous work, that it's related to the behaviors of the mouse is doing. Yeah, and also kind of, this is consistent with Nick's paper as well, that he didn't find these choice-related signals in visual cortex. But Kenneth is here, and he can correct me if I'm wrong.
So Carsen, you have about 5 minutes left. There's a follow-up question from Tony about timing, which I think is kind of at the heart of his question. But if if you have more things to go through, we can table that until the end.
Yeah, no, I'm pretty much done. So I'm just going to show this slide, and then we can go to the questions if that's okay. Sorry, I'm a little over. Just basically that there's clearly a lot of information in visual cortex. It doesn't seem like this information is necessarily any variability in encoding in visual cortex is necessarily driving behavioral variability. It seems like it's very much the case that I don't know. I'm being too strong here, but it's like the visual cortex has kind of in a high fidelity way encoding the external world, and then downstream decoders have to decide what to do with that information in order to navigate or do other visual tasks. And so we think of things that mice might use orientations for that might not be orientation discrimination, could be depth perception. They could use that information for that, for object tracking or object segmentation or other things. And in summary, we found that the error is very low in visual cortex. Again, this high fidelity coding. We don't know exactly why mice aren't using this information during behavior, but it's an open question. And I think a lot of people here and in other places are kind of working on how mice make decisions and use information, sensory information to make decisions. And I'd like to thank my collaborator and former advisor, Marius Pakitsariou, and also Mihaly Mihailos. He did all of the recordings that I showed today. And also we had a lot of technical support from people at Janelia. It's a great place to do research. And also thanks to my former collaborators and advisors at UCL. Next time it's Matteo Carandini, Kenneth Harris, and Chara Reddy. And the paper is on bioRxiv, the data is on Figshare, and the code is available on GitHub. And also, if you're interested in doing a postdoc or becoming a student at Janelia, feel free to reach out to me. If you're interested in working on kinds of these questions of how the mouse does complex high dimensional computations. Yeah, that's it. Thanks.
Thank you so much, Carsen. So we had a very active Q&A session. Please add additional questions in the chat. I guess the first one that's been upvoted is just, Tony, I mean, you could tell us if she answered it already, but I think the question about feedback versus attention in visual cortex was about kind of the timing of that signal. Can you elaborate on that at all?
Yeah, so that's a good question. So there is modulation by arousal through through the whole trial, I should say. But then this specific modulation that's corresponding to left versus right is more significant when the mouse is actually moving the wheel. And I don't have that figure actually in my presentation, I'm sorry, but that's like where basically taking the average of the neural activity in that subspace. But yeah, this is a little different because this is trained on the correct trials on left versus right stimuli, so it won't yeah, so there you can see that this is slightly smaller, but it's not directly showing what you're asking for. It's not trained on the choice, like for instance from the incorrect trials.
Okay, I think we're inviting Tony on screen, although I don't know if there's a delay. Great.
I think I need to find that figure, sorry. Hi.
Hi. No, I realized I wasn't doing very well typing. By the time I typed, you had gone on to a point which made my previous typing obsolete. So let me just, the fact that there is less information about choice in primary visual cortex, probably not a surprise, right? But the questions I have are questions about, first of all, where you think it comes from. It is there. So is it related to feedback from motor acts? Is it related to, as for example, somebody like Bruce Cumming might argue, it would be related to attentional factors in the case where the mouse was actually doing an orientation discrimination? Or is it related to something else? You've sort of touched on that. So maybe but I'd be interested to know if you know there, whether you say much about the time course with which the choice information arrives in visual cortex during the trial. And the second question, which I'll just tail on and then I'll abandon my little video slot here, is you can argue quite correctly on the basis of this that there's less information about choice in visual cortex than there is downstream. But is there still quite a lot? I mean, you've got a ton of information about orientation. Even if you have a very small amount of information about choice, that may be plenty, for example, to predict choice with almost perfect accuracy, given the number of neurons you have. So I wonder if in absolute terms compared how much information there was about choice in the different areas you looked at, rather than comparing the relative information about choice and stimulus. That's a mouthful. If it was unclear, let me know. But
no, those are both good questions. So I guess I'll address the first one, which is which is attention versus motor feedback. And so because this is a two-choice task rather than a detection task, I think the attention becomes less related to the choice of the mouse. Is the first thing I'll say. But there is attention. The activity across the brain is correlated with arousal throughout the trial. But then this choice information is more prominent and we think is then related to motor feedback after the mouse starts moving or during the movement period. So that's why we think it is related to motor feedback from different areas to visual cortex. We don't know from where, but that's our hypothesis. Similarly with our previous work, there's this time delay of around 40 milliseconds when you're trying to predict spontaneous behaviors that are represented in visual cortex. The activity is after the behavior by around 40 milliseconds. So there's kind of this delay that it could be afferent feedback or something, or proprioceptive feedback or something else has this time delay.
okay, good, thanks.
And then the second question, if I can remember it now, was about how big these signals are. And I wish I had that figure here. I'm sorry, I don't. In that so these are much smaller recordings, I should say too, that Nick did. They're still very big, but because it's neuropixels, you can't record as many neurons as you can with calcium imaging. So we couldn't perfectly decode the stimulus or the choice trial to trial from his recordings like we could from the calcium imaging, because there's not as many trials or as many neurons. But yeah, so you are able to better decode the stimulus than you are able to decode the choice, particularly in this period. I'm not sure. I'm not sure if that's the case directly. I haven't directly done that here, I don't think. So but I know for sure here that that's the case.
There's another question in the Q&A from Dima Rinberg. So it says, following Tony's question, what are the timescales of neural signals and behaviors such as the jumping squirrel? What are your arguments that the timescale you are studying is relevant to the task? And then the second question is, what is the role of the synchronicity between neurons, which is not taken into account in these considerations?
Right. Yeah. So that's a good question, and that's actually what motivated us to do these recordings here with these very short stimuli and to see that the discrimination thresholds were similar even if we showed 100 millisecond stimuli. I mean, that's still not totally biologically plausible. Like the mouse and the squirrel will be moving their head a lot while they're exploring, but it's more reasonably within kind of the time frame that maybe they would stop moving for a second or so. But yeah, I think things in general become more complicated when you think about time, and that's something we can't really resolve with calcium imaging recordings. Like, we don't have the time resolution to be able to decode natural movie stimuli, for instance, unfortunately. And then there are a lot of other interesting questions you could ask with those, and how well you can decode temporal stimuli as well, that we can't answer with this kind of technique so well.
There's one in the chat as well as one in the Q&A. So, there
was one other question about the synchronicity across neurons. And I think we do address that in the sense that, I mean, if there is synchronicity, we're able to decode despite that synchronicity. I think the mice are always kind of in a state where they're not largely in up and down states so much, like they're not super passive, but we can't really tell. We're recording too slowly to really tell the exact behavioral state. Like, we can't see up and down states in calcium imaging like you can in ePhys, unfortunately. So I can't say for sure if that's true, but yeah.
Really quickly, this is a question from Nick W. So it said, have you looked into the possibility of enhancing these specific neurons responding to orientation by also using optogenetics to see what effect this could have on the performance of the mice in the visual discrepancy task? It could be the small 1 to 2% decrease ends up being increased exponentially down the line, leading to the broad choices.
Yeah, I'm not sure exactly how, I'm not sure if you were doing a discrimination task, I guess you could activate all the neurons that choose one direction versus another direction. And I think people are doing those studies to see if that enhances and changes the performance of the mouse. And I don't know if that's going to be the case or not. Like if there's enough connections downstream that by chance, like if you increase their firing rate, that's enough for you to learn a decoder better. I would hope so, but I don't know what the exact limitation is, so I can't say whether or not that experiment would work.
Okay, thanks. I think the last question is from Noga Vardy in the chat, which is, what about lack of motivation? I presume that's the mouse's motivation on the task.
Not my motivation to decode the stimuli. We didn't really look at how motivation changes. We didn't look at this decoding across trials, across time. So generally the mouse will get less motivated the longer they're doing the behavior, and we haven't looked at that. But I mean, Nick's data is all online and published, which is great. So you can also play with these data sets too. Nick Steinmetz, by the way, sorry. I should say.
Okay, and I think the last question is from Shai Shoham, which is, when looking at the small stimulus example, did you select or exclude neurons with the non-overlapping receptor field? What % of the neurons are responding?
Yeah, when looking at the small stimuli, yeah. So we are in all of these recordings here, we're recording from a specific part of the tissue that has receptive fields. We're recording in depth, and we're only recording it within an area that has receptive fields that are over this small receptive, that on average their receptive fields are within this small stimulus. That's what we ended up doing. We didn't exclude any neurons. We just basically take a block of neurons there and use those to do the decoding. In terms of the % of neurons that are responding, that's kind of hard to say in calcium imaging. Yeah, I don't feel confident enough to put a number on that. I think I'd want to look in the EFIS data to say definitively what % of neurons are responding. I mean, it's going to be on the order of a fifth or so of the neurons in general, like, or maybe a little more, it depends. But it is interesting when you do these recordings, like if you show different contrasts or drifting versus non-drifting gradings, that different neurons actually are responding to those stimuli. And you can look at that, for instance, in the Allen Institute data too. And so there are different sets of neurons responding to different sets of stimuli, for sure, which is also interesting, but not something we looked into.
Great. Well, if there are no other final questions, I'd like to thank Carsen again for a really interesting, wonderful talk. And so thank you for joining us, and thank the audience for being very engaged and making it fun for everyone.
Yeah, thanks so much.